من محاضرات مادة الميكروبيولوجي في هذا العام الدراسي، ونتمنى لكم إن شاء الله الموفقية والنجاح. Now, our lecture is an introduction about microbiology. Microbiology is a science that deals with the study of organisms which are able to be seen by naked eye, such as bacteria, viruses, and others. Medical microbiology is a branch of microbiology that deals with microorganisms that causes the disease for human and other organisms, and can be divided into bacteriology, virology, parasitology. This means the branch protozoa, not elements, mycology and immunology. Microorganisms belong to kingdom microorganisms belong to the protista biological kingdom, including some eukaryotes, such as parasites, and prokaryotes, bacteria and archaea, viruses, viroid, and viruses. These are classified according to their structure, chemical composition, biosynthetic, and genetic organization. Now, prokaryotic cells, including bacteria, these have no organelles, no membrane enclosed the nucleus. This means there is no true nucleus and no histone. As you know, DNA of eukaryotes have a protein called histone. This histone gives supporting for DNA. But in bacteria, the histone is not a presence. Also, genetic material presence in one chromosome. This chromosome is circular, while in eukaryotic cells, the chromosome is linear. Also, they contain complex phospholipids, sphingolipids, and sterol. The ribose of these organisms or groups is 70S, while in eukaryotic cells is 80S. The most important feature of bacteria is the presence of cell wall that composed from peptidoglycan containing uramic acid. This is a unique feature for bacteria, while the eukaryotic cells has no cell wall. This is only the plasma membrane, except for some plants. Chromosomes, single, circular, and haploid, are short-lived unprocessed mRNA. Usually, RNA is not processed because the DNA of prokaryote has no intron and not present inside the nucleus. So the mRNA is not processed, while in eukaryotic cells, the mRNA is processed by several mechanisms, including gapping, polyadenylation, and splicing of intron. Also have coupled transcription and translation. In the cytoplasm, in a specific region called the nucleoid, the mRNA, when produced, is entered to translation. After transcription, entered to translation directly. Not required for processing, not required for passing through the nuclear pool, as present in the eukaryotic cells. Now, how can we classify bacteria? Bacteria business as a typical bacteria. This bacteria contain all prokaryotic structures, such as cell wall, plasma membrane, presence of cytoplasm, and others. Mycoplasma is the smallest type of bacteria characterized by its lack the present in mycoplasma. So any antibiotic that act on the cell wall by inhibition of cell wall formation is not active against mycoplasma, such as penicillins, cephalosporins, and vancomycin. All these antibiotic is not effective against mycoplasma. Rickettsia organism, this is the smallest bacteria and unable to grow outside the host cells. This bacteria is obligate intracellular bacteria and can beguile their ATP from host cells. Chlamydia, characterized by the presence of developmental cycle composed from two stages. Reticulate body is presence inside the host cell. Also, chlamydia is intracellular bacteria. Now we study the Koch postulates. These four important postulates by Koch to explain or define the causative agents of disease. The organism, the first postulates, the organism must always be found in a human with infectious disease, but not found in healthy one. This is clear. Also, the organism must be isolated from a human with infectious disease and grown in pure culture. This means the organism, يعني الشباب هاي لازم تفهموها. النقطة الثانية محمد عبد الباري. محمد صوتي واضح؟ محمد
دكتور واضح.
نعم دكتور واضح الصوت.
فأريد تنتبهون على هذه النقطتين اللي هم المهمات اللي 2 و3 اللي هي انه الكائن المسبب للمرض must be isolated from human، يعني لازم نعزله in pure culture from human with infectious disease. هذه الفقرة صارت عليها يعني تقريبا بعض التعديلات وبقت خاضعة للنقاش. This because of some organism cannot be isolated in pure culture, such as Treponema pallidum that causes syphilis. This bacteria unable to growing on culture media because of this bacteria is fragile and require and have the long generation time. The generation time of this bacteria about 29 hours. يعني شباب هنا مثلا عندنا احنا الايكولاي خلال 20 دقيقة البكتيريا الواحدة ممكن انه تصبح 2 وبالتالي ممكن عزلها in pure culture، بينما التريبونيما باليدم تحتاج الى 29 ساعة حتى البكتيريا الواحدة تتضاعف الى خليتين وبالتالي تحتاج الى وقت طويل جدا، اضافة الى انه هذه البكتيريا هي fragile. The third postulate is the organism isolated in a pure culture must be initiate disease, the same disease, when re-inoculated into susceptible animals. And fourth, the organism should be re-isolated from the experimentally infected animals. Now we study the structure of bacteria. Cells of bacteria is about 0.5 to 2 micrometer in diameter. They have not organelles, no nuclear envelope, have a single chromosome and sternate cell wall. Shapes of bacteria, cocci or round, such as in the genus Streptococcus, or clusters such as in Staphylococcus. The second is bacillus. This may be present as a single, such as in enteric bacteria that presents in the intestinal tract of human and animal, which presents as a normal flora, and some of this bacteria is true pathogen, and or chain such as lactobacillus, or coccobacilli, ovoid in shape, such as in enteric bacteria, or curved or coma shape in case of Vibrio, or spirillum in case of spirochetes. Bacterial structure the bacteria composed from flagella. Some bacteria are non-motile, so not contain flagella. بالله نسميها الأهلاب، أهلاب وليست أهداب. سيليا راح تشوفوها بالمادة الباراسايت. سيليا هي أهداب، هنا البلاي معناتها أهلاب. Capsule, plasma membrane, cytoplasm, cell wall, lipopolysaccharide in case of gram-negative bacteria, teichoic acid, inclusions, and spores. Flagella help in motility and can be considered as a virulence factor because of help in the spreading of bacteria from the site of infection to the healthy tissue. Some bacteria can be as a fast motile, such as a Proteus, which can be spread to entire of Petri dish. Arrangement of flagella is basis for classification. May be present as a monotrachus, presence of one flagella, or Lophotrachus is a tuft at one end, مجموعة من الأصوات تكون في إحدى نهايات البكتيريا. Amphitrachus presents in both end, or Polytrachus all around bacteria, such as in case of members of Enterobacteriaceae, E. coli, Klebsiella, and others. This figure explains the type of flagella and arrangement. Villi, short protein appendage composed from protein called pillin, smaller than flagella, adhere bacteria to surfaces. Villi is important virulence factor because of it help in the adhesion to the tissue and other surfaces, and antibody against villi block the adhesion. The F-billus used in conjugation to exchange of genetic information. But important of this structure is adhesion to surfaces and tissue. Capsule or slime layers, glycogalax or polysaccharide on external surfaces adhere bacteria to the surfaces and prevent phagocytosis. Complement cannot penetrate this sugar. Capsule is a polysaccharide composed from sugar. All bacteria has a polysaccharide sugar, carbohydrate in nature, except for Bacillus anthracis that causes anthrax. The capsule of this bacteria is composed from protein. يعني هاي تعتبر صفة مميزة لبكتيريا الباسيلس انثريسيس اللي تسبب الانثراكس اللي هو الجمرة الخبيثة، انه هي البكتيريا الوحيدة اللي تكون الكبسول مالتها مكونة من بروتين وليس كربوهيدرات، اما البقية فمكونة من كربوهيدرات. As prevent phagocytosis and complement cannot be penetrate this sugar. Also, capsule help in the adhesion of bacteria to the surfaces. Cytoplasm of bacteria composed from 80% of water, 20% of salts and protein. How an osmotic shock important. DNA is a circular haploid presence inside the cytoplasm in a specific region called the nucleoid. Also, plasmids is present is the extracircular DNA may associated with antibiotic resistant. This plasmid is different from the circular haploid chromosome. No organelles is present such as mitochondria or Golgi apparatus. Now, bacterial genome is not surrounded by a nuclear membrane, nor does it contain mitotic apparatus, is generally called the nucleoid or nuclear bite. This consists from polyamine and magnesium ions bound to negatively charged circular supercoiled double-stranded DNA, and the presence of a small amount of RNA polymerase, and other proteins. But the distinct feature is not the presence of histone. هذا الفرق الأساسي اللي بيناتهم بين البروكاريوت والإيوكاريوت، بين البكتيريا والإنسان، يعني إنه ال DNA مالت البكتيريا يكون circular double stranded DNA، بينما الإيوكاريوتيك is linear DNA. An eukaryote contain histone. Prokaryotes not contain histone or protein. Gene in eukaryotic cells composed from exon and intron, while in a prokaryote is composed from exon only. Bacterial ribosomes have a sedimentation coefficient of 70S and are composed from 30S and 50S subunit containing 5S, 16S, and 23S rRNA respectively. Ribosomes are the site of action of many antibiotics. طبعا معروف انه ribosome help in the formation of protein. So this structure is a site of action of many antibiotics that inhibit protein synthesis, such as aminoglycoside, gentamicin, amikacin, or macroids, erythromycin, tetracycline, and others. Now, most important cell membrane is a bilayer phospholipid. Water can't penetrate, flexible, not strong, ruptures easily, but the bacteria is a cell wall, give a rigidity for its structure. Osmotic pressure created by cytoplasm. This figure explains the structure of plasma membrane composed from hydrophilic region and hydrophobic, which has a fatty acid and glycerol. Because is bacteria not present in eukaryotic cells. Cell wall composed from peptidoglycan polymer, composed from amino acid plus sugars. Unique to bacteria, sugar containing N-acetyl glucosamine and N-acetyl gluramic acid. D form of amino acid used, not all form. Hard to break down D form. ال D form يكون غير حساس للإنزيمات الموجودة عند الهوست، لأن أغلب الإنزيمات تحلل ال form shape of amino acid, while D form is not. This enzyme is not effective, so hard to break down this D form and help in the protection of bacteria from host enzyme. This structure of peptidoglycan layers. According to peptidoglycan and thickness of peptidoglycan can be classified bacteria according to a specific stain. This stain called a gram stain. Gram stain divided bacteria into two important groups: gram-positive bacteria and gram-negative bacteria. Peptidoglycan layer in a gram-positive about can be reached to 40 layer, while gram-negative bacteria range between 4 to 8 layer. This structure of gram-negative bacteria cell wall is composed from periplasmic space that containing peptidoglycan and outer membrane containing phospholipids, lipoprotein, proline, lipid A, and O polysaccharide. Lipid A and O polysaccharide is a toxic component of gram-negative cell wall because of its able to elicit undesirable immune response and produce high amount of cytokine, such as in case of salmonella. Salmonella typhi is a bacteria that causes typhoid fever or enteric fever. The main virulence factor of this bacteria is the O polysaccharide chain and lipid A. That cause cell wall determine the shape of bacteria, strength prevent osmotic rupture, 20 to 40% of bacteria, and unique to bacteria. Some antibiotic effect directly on cell wall by inhibition the formation of peptidoglycan, such as beta-lactam drugs. Beta-lactam drugs including penicillins, cephalosporins, carbamazepines, such as penicillin, amoxicillin, ampicillin, cefotriaxone, cefotaxime, and others. Thick acid presence in gram-positive only, composed from glycerol, phosphates, and ribitol. Help or in the attachment of phage to the bacteria. This a cell wall of gram-positive bacteria containing thick acid and lipotic acid inside that enter to the peptidoglycan layer. Lipopolysaccharide, the functions of it is toxic, kill mice, pigs, and human. G. Septicemia, death due to lipopolysaccharide, such as in case of salmonella. Viral gene causes fever. DBT vaccine always causes fever. Adjuvant stimulates the immunity. شباب واضح؟ محمد.
اي دكتور.
واضح دكتور.
لحد الآن اكو سؤال؟ Now we study endospore. Few bacteria have a spore as a structure, such as in the genus of Bacillus and members of genus Clostridium. Bacillus is aerobic bacteria, while Clostridium is obligate anaerobe. Endospore help in the resistance of bacteria to heat, radiation, dehydration, cold, and boiling. This bacteria, such as Bacillus anthracis, can remain live for about more than 1 hour. The location important in classification. The spore may be central, subterminal, or terminal. The bacteria Bacillus styrothermophilus spores used for quality control of heat sterilization equipment by autoclaving. طبعا هذه البكتيريا هي بكتيريا غير مرضية. Classify spore according to location. In case of genus of Bacillus, such as Bacillus anthracis, the spore is present in the center of bacteria, while Clostridium may be present central, terminal, or subterminal. Now the differences between prokaryotes and eukaryotes. Prokaryote has cell wall, teichoic acid, lipopolysaccharide, endospore, circular DNA, plasmid, while all this structure is not present in eukaryotic cells. شباب اكو سؤال؟ انتم طبعا هذا القدر من المحاضره هو المطلوب من عندكم. امكانيه التفريق بين شلون تفرق بين الايوكاريوتيك سيلز and prokaryotic cells, the function and importance of bacterial structures. Each structure was the function of Koch postulates should be understanding the Koch postulates. شباب اكو سؤال؟ منو عنده سؤال؟ مصطفى.
نعم دكتور.
عندك سؤال؟
لا واضحة إن شاء الله.
المحاضرة واضحة؟
واضحة إن شاء الله دكتور.
هسه عرفتوا احنا شنو المطلوب من عندنا؟ أهم شيء إنه احنا نعرف نميز بين البروكاريوتيك سيلز والإيوكاريوتيك سيلز والوظائف الخاصة بكل. المحاضرة القادمة عن الأنتيبايوتيك نعرف إنه كل جروب من الأنتيبايوتيك والستراكشر اللي راح تشتغل عليه أو تثبط تكوينه. أبرار.
نعم دكتور.
واضحة إن شاء الله؟
إن شاء الله دكتور.
إن شاء الله. شباب الباقين
واضحة دكتور؟
ماشي إن شاء الله. طبعا أنا أشكركم شكر جزيل لحسن الاستماع إن شاء الله، وإن شاء الله يعني يسهل إنه تكون هاي المحاضرة فاتحة خير لكم، وأتمنى لكم الموفقية والنجاح. شكرا جزيلا.
شكرا جزيلا دكتور، تسلم.
شكرا.